Hello, everyone, and welcome to today's keynote presentation, Linking Cellular Location and Patient Prognosis in Brain Tumors. I am Jen Woods of LabRoots, and I will be your moderator for today's event. Now let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We will answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. I now present today's speaker, Dr. Rebecca Ihrie, Associate Professor of Cell and Developmental Biology at Vanderbilt University. For a complete biography on Dr. Ihrie, please visit the Biography tab at the top of your screen. Dr. Ihrie, you may now begin your presentation.
Hello, I'm Rebecca Ihrie from Vanderbilt University. I'm excited to tell you today about how obtaining and analyzing per-cell protein data has led us to some new understanding in brain tumors, an area of particular interest in my lab. So I'm going to tell you about a few stories today, one about identifying cancer lineage cell phenotypes within a brain tumor, and then a few stories at the end about understanding the immune component of those tumors and where particular cellular features are located within these heterogeneous tumor masses. But first, I thought I'd introduce why are we interested in using mass cytometry and how has it been particularly informative for us in glioblastoma. So one of the reasons that my lab became interested in using this approach is that when we think about everything that's going on within a cell, DNA and RNA profiling, which have been widely used in brain tumors and many other tissues, are certainly very informative. But when we think about the repertoire of things that a cell is capable of doing, this information is necessarily incomplete. And so one of our goals was really to complement what we had learned and what the field had learned at the DNA and RNA level to better understand beyond copy number analysis, sequence analysis, and transcript analysis, some of the things that we really wanted to see were how cells are dynamically responding to their neighbors and what their signaling capability at the protein level, shown here, really is. So many of the things that I'm going to show you, some of the important features for us in identifying cells that were of interest are things like signaling cascades. So phosphorylated signaling proteins, which are shown here in green with a red modification. And so, as you'll see, many of the stories that we're interested in center around things that need to be measured at the protein level. In my lab, we are particularly interested in studying brain tumors, and in the research I'm going to tell you about today, we have a special focus in the most malignant form of primary brain tumor, glioblastoma. So glioblastomas are by far the most common tumors that present in the brain that have origins within the brain. They're about half of all brain tumors and the huge majority of malignant brain tumors. Unfortunately, although they've been a heavy focus of study for many years, glioblastoma prognosis has not improved much over the past decades. What I'm showing you at the left here is the last major change to standard of care for these tumors, the introduction of the alkylating agent temozolomide in addition to radiotherapy. And while use of this agent does somewhat extend survival, as shown in this Kaplan-Meier plot on the left, you can see that this curve still trends downward. And so many patients have a very poor prognosis. And there's been a huge effort to try to better understand these tumors so that we can come up with new ways of targeting them. In particular, there's been an extensive amount of work to profile DNA and RNA changes within these tumors, resulting in their organization into molecular subclasses that are thought to reflect some of the underlying biology in different patients' tumors. And so there are currently 3 major subtypes of glioblastoma. They're distinguished majorly, primarily by alterations in growth factor receptor gene copy number, as well as transcriptional signatures that resemble different types of progenitor cells within the brain. However, one ongoing challenge in the field is that, as illustrated by the smaller survival plot in the bottom right, there are not large differences yet in clinical outcome between patients that have these different subtypes of glioblastoma. So more work needs to be done to understand how we can couple these biological variations and different subclasses to features that might be targetable and might improve outcome. And there are several reasons why it's been a challenge to transition these classifications into understanding that we can use to design new therapies. One of them is a problem that is common to many solid tumors. Glioblastomas, as illustrated here in the cartoon, are complex tumors. So they have many different cancer lineage cells within them, cells that resemble different stages of neural stem cell differentiation, but have cancer lineage mutations in them. These tumors are also highly infiltrative, and so there are many normal cells within any resected tumor mass that we study. For example, neurons, astrocytes, and glia, the resident cells of the brain. There are also endothelial cells. These tumors are noted to have abnormal vasculature, and importantly, there are many different types of infiltrating immune cells. And so many of the analyses that I referenced before use bulk DNA or RNA profiling, which, while informative, are difficult in allowing us to see the cell-to-cell heterogeneity within these masses. And this is perhaps easily illustrated in one example piece of data from our group. So these are cells from a glioblastoma tumor, which underwent copy number alteration analysis. So this would be considered potentially a classical subtype of glioblastoma, and based primarily on having an estimated copy number of the EGFR gene of 22 versus the normally expected 2. However, when we look at this biaxial plot of S100B, an astrocyte marker, and EGFR, the protein that we would expect to be highly expressed across this tumor, when you look at this contour plot of all of the events where we measured EGFR protein expression on the cell surface, what I hope you can see is that although there are certainly some cells that express EGFR protein, the huge majority of cells in the tumor actually don't have very high protein level expression. And so again, copy number alterations, while informative in helping us think about the biology of the tumor, haven't told us the whole story yet. So some of the things that we are interested in understanding in my lab, and the reason that we developed the tools I'm going to introduce today, are that we wanted to understand these heterogeneous mixtures of cells and also how they vary across patients. And so one of the driving questions that has interested my lab and collaborators over the last few years are, one, how can we find highly malignant phenotypes across glioblastoma? So how can we find the most aggressive cells, the ones that are driving recurrence, which might not be the majority of the tumor mass, and whose abundance might vary across patients? And also, how can we do discovery work in smaller cohorts of patients? So for example, clinical trials might require hundreds of patients, but for discovery work in biology, we might need to work with smaller groups of patients. In the study I'm going to show you, we looked at a discovery cohort of 28. And so how can we leverage all of the information that we get from approaches like mass cytometry, where we have millions of cells and lots of data on specific parameters that we measured, to figure out which of these parameters help us find the most bad actor, the cells that are driving the worst outcomes, and then identify their targetable features. So the approach that we elected to use as a complement to many studies using single cell sequencing at the DNA and RNA levels is mass cytometry, or CyTOF. And one of the reasons that we were particularly excited to use this is that it allows us to measure many features. We selected a panel of about 40 different parameters based on our knowledge of brain tumor biology and some of the previous genetic analyses in the field. And it lets us look at millions of cells. So we're really well powered to try to find rare subsets that might be a minority of the primary tumor when it comes from the operating room to our lab. So in this cartoon schematic, I'm showing you a general workflow that we developed and published together with our collaborators in the Irish lab at Vanderbilt. In the study I'm talking about today, All of the patients had IDH wild-type glioblastoma, the most common molecular classification of glioblastoma. And all of these are primary tumors, meaning that this is before, this is from the initial resection before patients undergo temozolomide and radiation therapy. So when tumors are resected in the operating room, within an hour, they come to our laboratory and are viably dissociated and viably cryopreserved using protocols that we have since published, so that we have millions of cells archived in liquid nitrogen that we can then bring out and study through immunostaining and mass cytometry. So we first developed a panel of antibodies to allow us to find immune, endothelial, and other infiltrating cells, and then to focus in on the presumed cancer lineage cells that are the majority of the cells in these samples. And then, in order to try to identify cellular phenotypes in an unbiased way, we developed a workflow called RAPID, which I'm going to dissect for you in more detail now. So some of the reasons that we were really excited about RAPID are that, one, it couples the per cell data that we're able to obtain using mass cytometry to continuous features like things that we measure in the clinic. So for example, progression-free survival, which would be measured in days or weeks, and overall survival, which in glioblastoma still has a median time length of about 15 months. And this is something that was relatively unique to this tool. There are other tools for analyzing high-dimensional data, but they often require either that you have prior knowledge of the normal tissue lineage hierarchy or normal samples for comparison. This is often difficult to obtain, as samples of normal brain are not readily available in our case. Or the tools require that you in advance assign your tumors or other samples that you're measuring to specific categories. And in glioblastoma, we really wanted to take an unsupervised approach where we didn't know outcome before the cells were analyzed. So we wanted to analyze all of these samples together and then identify in an unsupervised way which cells were associated with bad outcome. And that is what RAPID does. So I will take you through the workflow in more detail. But in brief, as you can see on the bottom half of this slide, we took over 131,880 glioblastoma cells from 28 patients, used optimized clustering to assign all of those cells to clusters based on the parameters that we measured, and then asked which of those clusters are meaningfully associated with outcome. So if a patient has very high abundance of cells that fall into a particular group, is their outcome better or worse? And in this study, we were able to identify both glioblastoma negative prognostic cells, whose abundance is predictive of especially bad outcome, and glioblastoma positive prognostic cells, whose abundance indicates an especially long overall survival. So now I'd like to take you through how this workflow works and some of the interesting things that we found about the biology of these cells. First, we, in this case, wanted to computationally exclude immune and endothelial cells, not because we're not interested in them. I have a few bits of a story on the immune compartment of these tumors coming up in a minute, but because for this initial study, we were particularly interested in cells that are primarily cancer lineage cells. So after excluding immune and endothelial cells on a per patient basis, so here removing the orange, blue, and red islands that are marked on the right-hand part of the slide, we then took all the other events in each patient's map and concatenated them together, as shown in this cartoon format here. So here we took what we're calling glioblastoma cells, so non-immune, non-endothelial, but pooled the data together for all of the 28 patients with equal sampling from each. And then perform dimensionality reduction to give us the map that is shown on the right. RAPID then uses optimized flow-SOM clustering, so self-organizing maps, to identify cell populations. And this is another point in the workflow where we really wanted the choice of cluster number to be determined in an unbiased way, rather than having the researcher try to hypothesize how many groups of cells might be present in the sample. Because at this time point in these diverse tumors, we really didn't know how many phenotypes to expect. So one of the things that the RAPID workflow does is it iteratively tests different numbers of clusters in the flow-SOM algorithm and identifies the right number to use to minimize intra-cluster variation while still having representative clusters. And so in this case, we ended up with 43 different clusters detected by flow-SOM. RAPID will then test, as I said, for each of these clusters, whether its abundance in an individual patient and across the patient cohort is associated with a hazard ratio that is above 1, meaning that as the abundance of this population increases across patients, the risk of death is greater, or whether it was associated with a hazard ratio less than 1, which is shown in blue on the t-SNE plot on the far right, meaning that if the abundance of this phenotype in an individual patient's tumor is higher, the risk of death is proportionately lower. And so doing several iterative testing steps that I'm alighting here to test for stability of these clusters and reproducibility across multiple analysis runs, we identified, as I said, glioblastoma negative prognostic cells, which are shown in red on this map. And you'll notice are all located together on one side of the map. And glioblastoma positive prognostic cells, which are shown in blue and predominantly on the right side of the map. And what was particularly rewarding for us is that if we then, again, in an automated way, classify the patients in the cohort as having a high proportion of either GNP in red or GPP cells in blue, and then look at overall survival, you can see that indeed patients who have a very high proportion of the GNP phenotype in their tumor, as shown on the red curve on the left-hand side here, have a much shorter overall survival, meaning that their tumors are especially aggressive. And conversely, patients with a high fraction of GPP cells in their tumor, this is the blue curve on the right-hand plot, have better than average overall survival. And you can see this in the blue curve, which pushes out further to the right. So this was really exciting to us because these kinds of survival differences, as I noted at the beginning, have not been easy to parse out in other types of data at the genomic DNA or RNA level. So seeing something that stratified survival was very exciting to us because it gives us an opportunity to better understand the biology of these tumors and think about how to target them. When we look at the individual channels that were measured, this is a subset of the features that were identified as enriched on negative prognostic or positive prognostic cells, and then compare them against the rest of the sample, which is shown in the gray histograms below. You can see that the phenotypes of the negative and positive prognostic cells are different. Principally, we see a lot of expression of the stem-like marker SOX2 in negative prognostic cells. So that histogram is further to the right. And interestingly, they also co-express this protein S100B, which is typically a marker of non-stem, more mature cells. So these cancer cells have a very mixed phenotype in which they somewhat resemble stem cells, which are quite rare in the normal brain, but then also resemble mature cells. And this is something that we wouldn't typically see in this normal tissue. And as you look at the right-hand side of the slide and look at these histograms in red, you might also notice that these cells in the negative prognostic population are particularly distinguished by having high levels of many phosphorylated proteins. That is, they have very active signaling through multiple pathways downstream of some of the growth factor receptors that are commonly mutated in glioblastoma. Although interestingly, they don't have particularly high levels of many of the growth factor receptors themselves. So something is keeping these pathways relatively elevated, and now we would like to know what. Beyond looking at this in the per channel analysis of histograms, we were also able to use a statistic developed by the Irish lab at Vanderbilt, marker enrichment modeling, to put a human and machine readable label on each of these populations. So at the top, I'm showing you the label for the negative prognostic cells. And if we look next to the up arrow for total protein denoted by P, you can see that these cells, as I said, are especially enriched for S100B and SOX2. They're also specifically lacking some other important proteins that denote different brain lineages, as well as the epidermal growth factor receptor. And if we look at the signaling signature denoted by S lower down in the box, you can see that these negative prognostic cells are also specifically enriched for signaling through STAT3, S6, and STAT5. Suggesting that they have a pro-survival signaling program that is active. And conversely, when we look at the label for the glioblastoma positive prognostic cells, although these cells are quite enriched for the epidermal growth factor receptor, which was a very unexpected outcome, they are also distinguished by a general lack of phosphorylated signaling proteins. When we go back to look at individual tumors, and this is a feature that I really like of having the per cell, per patient data available to us, we can also look at individual tumors and see that they reflect some of these overall phenotypes. So for example, LCO2, which is on the left, has a very high proportion of cells that, as shown at the top in the t-SNE plots, cluster on the left-hand side of the group map, so over in the negative prognostic area. We can also quantify this in the percentages that are shown at the right in a red box. And when we look at individual measured features, we can see in the heat on the t-SNE plots below that these cells in this patient are higher for many of the parameters that distinguish negative prognostic cells. And the converse is the case for W11 at the right. And if you look at PFS and OS, which are shown near the top, the outcome for each patient, you can see again that the negative prognostic cells being abundant is generally a negative indicator for overall survival. Interestingly, as I mentioned at the beginning, although we excluded the immune cells from the rapid analysis in the discovery of GNP and GPP, we of course were then interested in seeing whether the cancer lineage or glioblastoma cell features related to what we saw in the immune compartment. And one interesting observation right away was that glioma negative prognostic abundant tumors, so GNP high patients shown in red at the left, have a lower percentage, of CD45 positive infiltrating immune cells. So immune cell abundance is much lower in the negative prognostic cells, raising many interesting hypotheses about what the interaction between this cell phenotype and the immune response to the tumor is. And conversely, again, glioma positive prognostic high tumors in patients had a higher percentage of CD45 positive immune infiltrate. So having all of these machine-readable labels in hand and having all the high-dimensional data was fantastic for us for being able to do additional ongoing discovery work in these tumors and to understand these phenotypes. But we also wanted to think about how might this be translatable to approaches that neuropathologists typically use. And so one of the things we were very interested in testing is that now that we've been able to measure many parameters and identify the ones that are most selectively expressed on negative and positive prognostic cells, can we use that to find a more pared down way to stratify patients in a larger number of samples? So for example, can we use an imaging-based approach to then identify GNP-like and GPP-like tumors and still see effects on survival similar to what we saw in the high-dimensional suspension analyses of these solid tumors. So taking these labels on the left, marker enrichment modeling, and looking at the aggregate data shown on the right as a biaxial plot for EGFR and S100B, one thing that we noticed is that these two parameters are inversely correlated, both in the MEM labels and then when we look at the biaxial plot of the individual data. Samples tend to have a lot of EGFR or a lot of S100B, but fewer cells have both. And so we hypothesized that as these were the most enriched parameters, we might be able to use them to design a much more simplified staining protocol to identify tumors that had GNP or GPP-like cells. And so what I'm showing you here is analysis of a tissue microarray that had an additional cohort of 73 patients beyond the 28 that we studied in suspension. And across the top, you can see as we stained for many of the features that are enriched in GNP or GPP cells, that we were able to detect them and to classify patients as low, medium, or high for these parameters. Looking solely at EGFR and S100B at the bottom, we were able to design a stratification scheme based on the marker enrichment modeling labels that I showed you before to classify patients as GNP-like, GPP-like, or low for both. And looking at the right, if we then look at survival in those patients, looking at the red curve of survival over time, and then the blue curve, you can see that even using a different platform and a simplified approach, we're able to use the information from our CyTOF analyses to predict especially short or long outcome across the patients included in this tissue microarray. And so this has suggested to us that this can be turned into something that can be applied more broadly across large cohorts of patients, allowing us to think more about additional parameters. But of course, in my lab, the more numbers we can put on something, the better. And so the other future direction that we are just beginning and really interested to pursue is to try to analyze all of these parameters simultaneously on the tissue microarray. So one thing that those of you who do a lot of imaging might be considering as you look at this slide is that it can be very challenging across individual sections to then come up with a unified score for each tissue core on a microarray. So we have to do each one of the columns that I'm showing you in a separate imaging run, and then try to assign a score that we can put back with the scores for all the other parameters to classify the patient. So wouldn't it be nice if we could measure everything together? And that's something that we're working on now, together with the Bursky Center with Imaging Mass Cytometry at Washington University, St. Louis. And so one thing we're excited to do going forward is to take all the parameters that we used on dissociated tumors in suspension and apply them across this tissue microarray and see whether having more information gives us better classification of the patients included in the tissue microarray cohort. And as these data really illustrate, especially in my lab, some of the questions that we want to answer require spatial information. And so some of the things that I'm going to tell you about now focus on both tumor location within the brain. How does that affect the features of cancer cells and the immune cells within the tumor? And then how do we think about taking some of the phenotypes that we've discovered in suspension and figuring out where they localize within individual tumors or across individual tumors? And this is something that my lab has been interested in for a long time, perhaps because we study a niche that has a lot of specialized cytoarchitectural features. So for example, if you look at the right, the image on the right here, you can see this is the human brain stem cell niche with stem cells labeled in green and recently born young neurons in red. And simply, I think looking at this image brings home the point that these cells have very different shapes, which connect to their biological function. They're located in different parts of this niche. So some of them are right up next to the cerebrospinal fluid, the open space at the top. But they also extend a long process in green down to touch vasculature that underlies this niche itself. And so thinking about how these cells contact each other and how that influences their phenotype is going to be an important question going forward. And we knew that we wanted to investigate this because we had some clues from the clinical literature, including studies by a neurosurgery fellow in my lab, Akshay Kumar Mistry. So Mistry independently had become interested in this question of when glioblastoma presents in the brain, does it matter whether or not it contacts specific structures within the brain? And this was building on some earlier data in a small number of cases from studies at other institutions, but Mystery was able to build 2 cohorts of about 200 patients each to ask this question in a statistically meaningful way. And one of the things that he found that we were really excited about, so Mystery chose to classify patients into one of 4 categories based on their MRIs, and some examples are shown at the left. So about 50% of patients on their initial diagnosis, so when they first enter the clinic with glioblastoma, have a tumor that contacts the lateral ventricles. And these are the sort of black butterfly shape in the center of the brain. And it's an important area to us because one of the 2 stem cell niches of the brain is located next to the lateral ventricles. So about half of patients when they come in have a tumor that is contacting the stem cell niche, and half of them do not. In addition, a smaller subset of patients, as shown in the top right MRI, have a tumor that contacts the other major stem cell niche of the brain, the dentate gyrus. And so one of Mystery's key observations was not just to say, do patients have a tumor that contacts the lateral ventricle niche or not, but also to say, is there something special about the lateral ventricle niche versus the other stem cell niche? And to summarize quite a bit of work classifying MRIs on his part, what he was able to show is that, as shown in the survival curve on the right, if a patient's tumor contacts the lateral ventricle stem cell niche, but not the other one that is known to exist in the brain, those patients have shorter overall survival, so a worse outcome. And that's illustrated by the red curve in the Kaplan-Meier plot on the right. And he then did quite a bit of work to think about, well, did this connect to any other known predictors of outcome, such as the size of the tumor, the ability of the surgeon to resect it, or bulk DNA and transcriptomic profiles? And the short answer is that none of those things explain the difference that he has observed, raising some interesting questions about what biology might be behind the differing outcomes then. So is the stem cell niche or the cerebrospinal fluid next to it somehow influencing the biology of the tumor? And can we see this difference using some of the tools that we had already developed? Fortunately, in the viably cryopreserved cohort where we had done cytoph analysis, when we looked at survival relative to stem cell niche contact, we again saw the same pattern. So we already had a representative cohort in hand. And multiple people in my group, as well as the Irish lab at Vanderbilt, have begun exploring what might be different in the cancer lineage cells within ventricle contacting tumors, as well as the immune cells within them. So first, Justine Senave, a PhD graduate of my lab, was able to use an existing tool called Citrus to identify clusters of cellular phenotypes that are enriched in non-contacting tumors versus ventricle contacting tumors. And here, Citrus, in fact, worked very well for us because unlike at the start of the rapid analysis where we wanted to be unsupervised, here we had one clear criterion that allowed us to separate the tumors into 2 groups based on their MRI contact information. So we found a cluster of cells that is enriched in non-contacting tumors. And excitingly, the protein expression levels of cells that fall within this cluster very closely resemble the glioma-positive prognostic phenotype, suggesting to us that ventricular contact and the previously identified more favorable cellular phenotype might go together. And you can see this by comparing the Citrus results, which are in the top row. Cluster A is in purple and everything else in the sample is in gray. And then the individual histograms of positive prognostic cells from the prior study, the rapid study, beneath. And what I hope you can see is that the distribution of these features is very similar between the 2 analyses. And intriguingly, in addition to seeing some differences within the cancer lineage cells, when we look at the immune component of stem cell niche contacting or distant tumors, we also see some very interesting differences that suggest possible future directions in immunobiology. So this is the work of Todd Barkowiak, a shared postdoc between my group and the Irish lab at Vanderbilt. And in brief, Todd has analyzed the infiltrating immune component within all of the resected tumors that we had previously analyzed for features within the non-immune, non-endothelial compartment. So looking at the immune cells and plotting the data from multiple patients together, as shown across the top, Todd was able to compare all of the data together, as shown in the contoured Visney plot at the left, and then separate out only ventricle contacting tumors, which are in blue in the contour, the next contour plot, and non-contacting tumors shown in the red. And what I hope you can see when we overlay those plots, and then when we computationally seek for clusters that are enriched in only one or the other, shown at the right, is that there are some cells that are predominantly seen in contacting or non-contacting tumors, i.e. that there are areas where these maps don't overlap. And using rapid analysis, again, on these data, Todd was again able to identify clusters of cells with similar phenotypes, here shown either in red or in green, that are associated with especially good or especially bad outcome in patients. And I'm just showing you two examples here where patients are dichotomized based on whether they have high or low abundance for some of these immune cell phenotypes. And for example, when we look at subset 17, shown in red, and separate out patients that are abundant for that cluster, that would fall primarily in the stem cell niche contacting classification, we see that those patients do worse. So these data collectively suggest that stem cell niche contact has a really profound effect, both on the cancer cell phenotype and on the immune cell phenotype, and that going forward, we're really interested in finding ways to couple what we can see on MRI, which is a routine visualization that is done for glioblastoma patients, to information that might help us think about what treatments might work best on those patients' tumors. So going forward, I told you before that spatial information is important, and we just spent some time discussing how location within the brain has a significant influence on tumor prognosis, as well as the cells that are found within the tumor. But in addition, I wanted to take a moment to highlight where imaging mass cytometry and thinking about cell-to-cell variation and distribution within the tumor is starting to help us think about how particular phenotypes cluster together within a tumor, and how we can identify cell-to-cell variation and understand the biology and spatial organization of these tumors. So I have two example imaging mass cytometry cases, one again focused on glioblastoma, and then a second study of subependymal giant cell astrocytomas, a different brain tumor where stem cell niche contact and location is also very important, but we know less about the signaling variation within an individual tumor than we do about glioblastoma, and also less about its differentiation state. So to do this, we spent a significant amount of time in my lab developing and validating a brain-centered imaging panel. I'm showing you one example here where we made custom conjugates of an antibody against, for example, phosphovimentin, which is shown in red at the far left, and marks dividing progenitors in the early brain, as well as a subset of brain tumor cells that resemble these progenitors. So on the left, you can see the expected staining pattern. This is the antibody before we conjugated it to a metal. You can see overall brain architecture in blue, and then the merged image. And then we always, after conjugation to a metal, will restain the same control sample, and that's what you're seeing on the far right, to make sure that we see the same distribution we were expecting based on the original clone. So after some optimization and selection of parameters of interest based on our biological knowledge, we began staining with a trial panel that contains antigens that overlap with the rapid story that I told you about before, as well as some other lineage-specific transcription factors and phosphoproteins that were particularly of interest to us. And when we began staining glioblastoma samples, we got images like the following. So this is a single glioblastoma patient's case and a subset of the antigens that we tried. you can see phosphostat-3, tyrosine 705 in yellow. And one thing that you might notice is that we see what appears to be a non-random distribution of signaling here, where we might have clusters of cells signaling together. So we're very interested to use some of the tools developed by other presenters at this conference to push our understanding of that localization further, and then ultimately do some mechanistic testing of the hypotheses we get from these images. We also were really excited to be able to have lots of parameters simultaneously. So this is another set of antigens on the same piece of tissue. And this staining, this pilot staining was done at King's College London Guy's Hospital, so a special thank you to them. And some of the things that are of interest to us are that in addition to the clusters of phosphostat-3 that I previously indicated, you can also see that we see phospho-ERK in the bright magenta in many spots throughout the tumor, but again, located in clusters that we think will be interesting to explore when thinking about local signaling. We have been able to use tools such as histocat and then marker enrichment modeling to take these images and assign each segmented cell within the image to a particular cluster. And then for us, using marker enrichment modeling on these data is especially helpful in finding clusters of interest. So for example, when we take the image on the left, and then you analyze the data in histocat, giving us the dimensionality reduction plot at right, we can single out clusters like the one in dark red on the far right, cluster 13, and see in the MEM label in the listing at the right that this cluster, for example, is especially high for phosphostat-5. So phosphostat-5 is specifically enriched in that cluster. You can see that a little better here when we look at the heat for phosphostat-5 on the same map. Or on the original image. And we are particularly interested in these cells because phosphostat-5 is one of the driving markers of the negative prognostic phenotype in glioblastoma. So we're very excited going forward to think about distance from the stem cell niche and other features that can be really nicely measured using imaging approaches, and then taking those data and applying all the quantitative analysis tools that we've already developed for suspension cytoph. So in my last few minutes, I wanted to show you one of the other brain tumor types that the lab is really interested in studying. These are periventricular brain tumors, so they present next to the same stem cell niche I was talking about before, but these are a really interesting contrast point to highly malignant glioblastoma because they are benign and because they present very early in life in children with a disorder termed tuberous sclerosis complex. So these tumors are of interest in part because their origins are somewhat poorly understood, but also from a quantitative biology perspective, they're interesting because they're quite poorly defined. They're described as having a mixed glioneuronal phenotype, which essentially means that you can see markers of nearly every brain lineage in them. They're known to have high mTOR complex 1 signaling because tuberous sclerosis complex is a disease where regulators of mTOR are mutated. But beyond that, we have relatively scant information on how to distinguish them from other benign brain tumor types and other tumor types that present in the brain where mTOR signaling, for example, is also altered. So one of the things that we are particularly interested in doing moving forward is using both suspension and imaging mass cytometry to get a much more quantitative description of what the cells in these tumors look like, and again, what are some selectively enriched targetable features. So these tumors are relatively rarely resected, but fortunately through collaboration with the head of the TSC clinic at Vanderbilt, Kevin S., we do have access to some of these samples. And I'm showing you imaging of one of them here. So this is again, one subappendable giant cell astrocytoma tumor that's been stained with our pilot panel of antibodies. We're particularly interested in things like phospho-S6, shown in red here because it indicates high mTOR activity, as well as lineage markers like TTF1 or NKX2.1, which would tell us something about the region of origin of this tumor. If we look at an additional subset of the antigens, we can also see some other classic readouts of mTORC1, such as phospho-4ABP1, which is shown in red. And one of the things that we were interested to see right away is that, again, this is not evenly distributed throughout the tumor. We might hypothesize because these patients are carrying a mutation in a regulator of mTORC1 that every cell within the tumor might have high mTORC1 activity and that there would not be a particular distribution. But it doesn't appear that that is the case. Instead, if we take these images, segment them, and use histocad and MEM, as in the previous approach I was discussing, you can see that cells are assigned to clusters, here indicated by colors, that are not evenly distributed throughout the tumor. And what was particularly interesting to us is that when we look at some of our key markers of interest, such as, for example, SOX2, a marker of stemness, and phospho-S6, a marker of high mTORC1 activity, what we see, a little bit contrary to expectations in the field, is that while there is one cluster of cells shown in cyan that has very high SOX2 expression relative to everything else, it is actually not the same cluster as the one that has the highest mTOR complex 1 activity, which is indicated in red. So we're going to be thinking a lot about how do these two features, which were thought to generally be present in the same cells, actually relate to each other within these tumors. And interestingly, as we have gone back to close the loop into suspension mass cytometry analyses of samples from TSC patients, we see, again, a similar pattern. So in my last data slide, I'm showing you suspension mass cytometry, individual parameters at the top, and then the t-SNE analysis at the bottom with heat for individual markers. We've been able to go back in and see, while we indeed have a lot of cells within resected masses from patients that have a lot of SOX2 expression, as shown in the bottom right, those SOX2 high cells, or the cells that have the highest SOX2, are not actually the same events that are very, very high for phosphorylated S6, which is shown in the very bottom right here. And you can see when looking at the heat on the t-SNE plot that one island is very high for phospho-S6, but not for SOX2. And in fact, that upper island bears the marker of expression of a different lineage progression marker, SOX10. So some of the central findings that I've tried to highlight today are that particularly in glioblastoma, where transcriptomic and other genomic analyses have not yet allowed us to stratify patients into different groups based on outcome. Instead, measurements at the protein level, particularly including phosphorylation events, have helped us find prognostic subgroups of cells that we can investigate further. We did a lot of this through the custom workflow termed RAPID, which has now been published. But we can also, using the information from RAPID, come up with simple stratification assays and go back to other types of high-dimensional data and ask how our findings, for example, in fixed tissue, compare to suspension. Ultimately, we're very excited to find, too, that stem cell niche contact changes both cancer cell and immune cell features and is linked to some of the differences in prognosis that we found before. And importantly, these phenotypes are not uniformly distributed through the tissue. And so going back and forth between what we can discover in suspension and in fixed tissue has been an incredibly useful tool for us. So I'll conclude just by thanking the small army of excellent collaborators who joined in on this work, especially Jonathan Ihrie, the director of the Cancer and Immunology Core and the Mass Cytometry Center of Excellence, in addition to his own lab, our close collaborator, Vivian Gama, who's a stem cell biology expert, Kevin S., the head of the TSC clinic at Vanderbilt, as well as his colleague, Carrie Fu, the 3 neurosurgeons who worked diligently with us over years to collect patient samples, the graduate students and postdocs in the Ihrie, Irish, Gama, and S labs, as well as the excellent staff shown below, and the neuropathologist, Brett Mobley, and neurosurgery fellow, Akshay Kumar Mistry, who really took the lead on making sure that we had all the annotated clinical data needed. And finally, of course, I would also like to thank all of our funders, including the individual donors and patients and their families. And I will stop there and take any questions.
Thank you, Dr. Ihrie, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located in the far left of your screen. Let's get started. Our first question is, how do you know which ones are GPP versus GNP at the beginning?
Okay, can you hear me? Just let me make sure first. Great.
Yes.
Okay, so thank you for that question. So I think what you're asking is, how can we call individual patients or individual cells as being part of the GPP versus GNP populations, or whether a patient is called as low or high for GPP and GNP? Let me start with the cells. So one of the things that was really important to us in designing RAPID is actually that we wanted to be naive to a lot of that information when starting the analysis. And so we actually did not know what the important features for GNP and GPP were going to be until the analysis was complete. One of the things that we really like about RAPID is that you can essentially give it your patient CyTOF information. So however many patients you've included, usually around 25 is useful for starter cohorts. So you take the FCS files or the IMD files and feed the data into RAPID together with in this case, survival times for all of the patients. So you start without knowing, for example, which patients were good or bad outcome necessarily. And then most of the remaining steps are done in an automated fashion. So the GNP and GPP phenotypes and then the labels that define them are generated as part of the RAPID workflow. So the short answer is we did not know at the beginning which cell phenotypes and which features were going to be the most important ones. And that's where having a lot of parameters to be able to analyze was really helpful, because we were able to cast a very wide net. For example, we might have hypothesized based on the literature that expression of other stem-like features like CD133 would be very important for identifying prognostically meaningful cells. But it turns out we measured that along with 8 other things associated with stemness, and only SOX2 was selectively enriched on the GNP population. So I hope that helps answer something about the cells. And then in terms of how patients, for example, are assigned to being high or low for a particular cell phenotype, so do they have high abundance or low abundance, that is also done in a largely automated fashion based on the distribution of abundances within the cohort. So again, we try not to create an artificial cutoff. We try to find it in a statistically determined way.
Okay, thank you for that, Dr. Ihrie. Next question. Since GNP is inversely proportional to immune cells, does the same apply to GPP? In other words, is immune cell enrichment in tumors a good predictor of survival? If yes, how do you explain mesenchymal GBM's survival, which is heavily infiltrated with immune cells?
Right. So I can see the speaker as familiar with some of the GBM literature. So let me answer the GPP question first. So yes, GPP high patients have a somewhat proportionately higher immune infiltration, and that's just looking at total CD45 positive cells. So while it's somewhat went along with survival in our smaller discovery cohort, I think there's a more complex story, which you're getting at with the question about glioblastomas that are transcriptionally classified as mesenchymal are thought to have a lot of immune infiltration. And that's based partially, I think, on immunostaining studies of TMAs and partially on transcript level analysis. So I guess what I would say, and what I think some of Todd's data that I briefly covered demonstrated, is that although the proportion of immune cells in the tumor potentially has a small and has some influence on survival, it is definitely not the whole story. So it's much more important what the exact complement of immune cells within the tumor is. And beyond that, I would also say, just because T cells or specific subpopulations are present does not actually guarantee that they are capable of carrying out their, their normally intended function. So I think it's going to be really important to look at the ability of those cells to actually mount an anti-tumor response and some of the signaling pathways that would indicate that activation is happening. So I think while CD45 positive gives us a clue that something is going on, we need to explore it more deeply. So.
Okay.
And I'm sorry, can I also just add, the other thing to keep in mind is that when we try to look for protein level proxies of the transcriptomic classification, so for example, you would use transcriptomic or genomic alterations to call a tumor as mesenchymal or another subclass, we don't always see that perfectly matches expression of those things like CD44 or MET for the mesenchymal subclass at the protein level. And so I also think one of the things that will be really interesting as we go forward is to go back and retrospectively use transcriptomic and genomic classification on tumors that we know have GNP or GPP to try to figure out how those categories fit together. So,
okay. Our next question, I see HLA-DR plus CD45 minus cells. Mm-hmm. What are those in TSNE plots and CD64 plus CD4 plus cells? Can you please comment on that?
Yes. So Todd, the generator of that data, is actually in the audience, and I have asked him as well. So one thing that I think is important to point out about the particular TSNE plot in question is that all of the events that are on that plot have already been gated for being CD45 positive, i.e. we've already pulled out all of the immune cells. And so when we display the heat for 45, on that TSNE plot, they're going to look very low when in fact it's that the range is much narrower than you might think. So that particular population we think is CD45 low, HLA-DR positive microglia. And then on the other side, the CD4, CD64 are, we think are macrophages. And I'm sure I or Todd would be happy to talk about this more offline if you would like. So.
Okay, great. Next question is, which is the experience using any of these markers to follow patients after the tumor has been removed in order to check their prognosis?
So if we use any of these to follow patients, so all of the analyses that I showed in the rapid workflow are analyzing tumors at the initial resection. So this is the tumor is resected, then the patients, in this case, underwent standard of care treatment. So we do have information both about time to recurrence of the tumor, so progression-free survival, which I didn't have time to talk about, as well as overall survival. So in our case, and in what I was able to show today, we were able to test how well the combination of markers that identify GNP and GPP cells predict outcome for the patients. And when we did the tissue microarray staining that I showed a little bit later, the part of the point of that was to try to say, can we pull out some of the most highly enriched markers and use them to stratify outcome in an independent cohort, so trying to get more at this predictive question. What I think will be really interesting to do going forward that would take this a little further is to see when you look at recurrent tumors, do they converge on, say, a more GNP-like phenotype? So can we learn something about what tumors look like post-treatment, or which of these populations are especially refractory to standard of care or investigational treatments? So I think that's what the questioner is getting at, but please let me know if you have a follow-up.
Okay, we have time for one more question. Mm-hmm. How much time does it take to collect data from a core using IMC?
So I'm going to say that this actually I could probably talk about in the Ask the Expert session this afternoon, or Dr. Bodenmiller could. If you don't mind, I'd like to answer the one after that. I see,
yes,
is rapid easily translatable to other diseases and populations? Mm-hmm. And then a second question about, can you use it on single cell transcriptomics? So first, we specifically designed rapid because we wanted something that would be translatable to other diseases and cell populations. And in fact, in the paper, we actually tested it on one of Dr. Davis's, who spoke earlier this morning, one of her data sets, and we're independently able to find the same, some of the same phenotypes that they had shown were predictive of outcomes. So we know that it can translate to other CyTOF data sets quite well. And the intent is that it is modular and can be applied to lots of other single cell data types where you have a continuous measurement that you want to use. That could be patient outcome in days or weeks. It could be time to relapse. It could even be percent immune infiltrate. So the idea is that anything where you have that kind of continuous data and a sort of medium-sized investigational single-cell data set, you might be able to use this approach.
Okay. Thank you again, Dr. Ihrie, for your time and for your important research. Before we go, I'd like to thank the audience for joining us today. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. As a reminder, please visit the Fluidigm booths in the exhibit hall to interact with representatives and gather collateral into your virtual briefcases for later review. This webcast can be viewed on demand through September of 2021. Until next time, goodbye.